Hello everyone, welcome. So in this video, what we're going to be doing is we're going to be going through chapter 7D, which is looking at the adaptive immune system or the third line of defense. So hopefully you've watched my last couple of videos where we've gone through the first line and the second line, which is the innate immune system. So in this one, we're going to go through the adaptive immune system. So just a reminder of what the adaptive immune system is, we did touch on it. So if the first and second lines fail, so if our innate natural immunity fails, this is when that third line or the adaptive immune system is going to be activated. So the adaptive immune system is something that we are not born with. It's something that develops over time. So it will respond to specific pathogens, and in response to those pathogens, it will form immunological memory, and it's way more specific. And because of this, it's also quite a slow reaction. Because the first and the second lines have to fail, and then the adaptive cells have to be activated, and all these things have to happen, which we're going to learn about in a second, this reaction is quite slow. So that's why some people that have quite severe disease, it might last for several weeks, if not more, because the adaptive immune system takes about 2 weeks to activate. So cells of the third line are often referred to as lymphocytes. So the innate immune cells, hopefully you remember, were referred to as leukocytes or white blood cells. The third line is referred to as lymphocytes. This whole process is initiated by the presentation of non-self antigens to specific immune cells of the adaptive immune system. So hopefully you remember in the innate immune system, we already highlighted a couple of these antigen presenting cells. Macrophages and dendritic cells were the main two. So we're going to look at how that actually happens, so how they actually present those antigens to our adaptive immune system. So the two things that are really important to note here that differ from the innate immune system is specificity and immunological memory. So if you remember, the innate immune system is not specific. It'll just respond to anything. It'll respond to anything pretty much the same every single time. The adaptive immune system is really special because it is specific and it provides memory. So the adaptive immune system will only respond to specific pathogens in a unique and tailored manner. So there's only going to be, one specific type of cell that's going to respond to one specific type of antigen. So it makes it really specific and unique and tailored to whatever that pathogen is. The adaptive immune system is also going to provide immunological memory. So what that is because the immune system results in the production of lots of cells that they're called memory cells, which we'll go through in a second, they will sit dormant in the bloodstream or in the lymph nodes. And what's going to happen is if you get reinfected with the same pathogen, you're going to be able to basically destroy it a lot more quickly and a lot more effectively because you've got memory cells that are going to respond to that pathogen, which is obviously very important for our long-term immunity, and that's what forms the basis of vaccines as well. So we're going to go through two different types of adaptive immunity. So adaptive immunity is split into two different types. So we've got our humoral immunity, and we've got our cell-mediated immunity, and we're going to go through each of those. I'm going to go through this one first and this one second. We're going to go through each of those and see, why a certain pathway would be activated and what happens in that pathway. So we're going to start with the cell-mediated immunity. So this is involving things called T lymphocytes. So the adaptive immune system is split into B and T cells, and the cell-mediated response refers to the use of T cells to fight an infection. So cell-mediated immunity involves the destruction of infected or abnormal cells. So that's why it's called cell-mediated. So this immunity is based off, it is mediated by infected cells. So it will find cells and it will destroy them. This type of immunity is really, really good for intracellular pathogens. So if the body or if the antigen presenting cell recognizes an intracellular pathogen, so that's things like viruses that actually enter the cell, this is when normally a T cell or a cell-mediated immune response will be activated, because T cells are really good at targeting those intracellular pathogens because they target infected cells. So throughout this, we're going to be talking about three different types of T cells. This is the main effector cell here. So effector cell just means that this is the one that does most of the, killing, destroying most of the work. But we've also got these two cell types that are really important as well. So we've got our T helper cell. So in a minute, we're going to go through how antigen presentation works, and the T helper cell plays a major role in that. So this is a differentiated T lymphocyte that supports the functioning of a number of different immune cells, including the cloning and differentiation of T and B cells. So this is like the starting point. So an antigen presenting cell is going to go to a T helper cell first, and they're going to be the cell that's going to activate all of the other cells around it, basically. We then have our cytotoxic T cells, which I said is our main effector cell. So they're going to again differentiate from a T lymphocyte, and they're responsible for the destruction of infected and abnormal cells. So they're the ones that are doing all the heavy lifting. And then we have our T memory cells. So like I said, when a pathogen is discovered by the adaptive immune system, after the pathogen has been destroyed, these T memory cells are going to form, and they're going to remember that pathogen. They're going to remember that antigen. So they're going to provide long-lasting immunity if we get infected again. So let's go through this. So I've gone through it in 3 steps. So we're going to start with how all of this actually gets started. Then we're going to go through the clonal expansion process, and then we're going to go through what cytotoxic T cells do. So the first thing we're going to start with is antigen presentation. So this actually happens whether it's a cell-mediated or a humoral response. So whether it's T or B cells, this has to happen every single time, because the adaptive immune system can't respond to anything if it doesn't know that anything is there. So we've got our antigen presenting cells. So hopefully you remember from the last video, these are things like dendritic cells, macrophages as well can do this, and also B cells can also do this, which we'll touch on when we do our humoral response stuff. But the main antigen presenting cell that's normally going to do the adaptive immune system activation is going to be our dendritic cell. So these antigen presentation cells, especially the dendritic cell, are going to engulf and digest the pathogen, and they're going to display that pathogenic antigen on their MHC class II, which we talked about in our innate video. So they're going to present that antigen. Then what they're going to do is they're going to travel through the lymphatic system, which we're going to touch on a little bit later. So this is the main system that our adaptive immune system uses to move around. So they're going to travel through this to something called a lymph node, which is where all of our adaptive immune cells are sitting and waiting. So while they're in that lymph node sitting and waiting, they're called naive cells. So we've got naive T and B cells. So they're just sitting there and they're just waiting to get activated. So the dendritic cell is going to, and you can see it down in this diagram here, the dendritic cell is going to present that antigen to a T helper cell. So the T helper cell, hopefully you remember, is like the main activator cell. So that's the first one that's going to be activated, and then that T helper cell is going to go on to further activate other cells. So this is our T helper cell over here, this green cell here, and this is our dendritic cell. So you'll see that the dendritic cell has its MHC class II, and it's got the foreign antigen that's holding there. It's going to go to a lymph node. It's going to find the T helper cell that is specific for that antigen, because remember, there's only one that's going to be specific. And that T helper cell is going to bind to that antigen, and then selection is said to have occurred. So when a T helper cell becomes activated by this antigen presentation process, it is said to be selected. And then that selected T helper cell is going to go on to initiate either a humoral or a cell-mediated response, depending on what the pathogen is. But regardless, this happens every single time an adaptive immune response is initiated. So once that T helper cell has been activated or selected, if we're going to follow a cell-mediated pathway, the first thing that's going to happen is we're going to have clonal expansion. Cell-mediated. Oh, my thing's really slow. There we go. So this is the cell-mediated pathway, remember. So the clones of the selected T cell. So the T cell that has been selected, that T helper cell, is going to differentiate into 2 different types. So the 2 different types that we talked about. So it's either going to become, or it's going to activate T cells to become cytotoxic T cells or memory T cells. So you'll see here, so after this antigen presenting cell has, done its job, it's going to go on and it's also going to activate naive T cells as well. And the T helper cell is also going to play a part in this. So once the antigen presenting cell has presented to the T helper cell, it's going to go off and it's going to present to other naive T cells. The T helper cell is going to help by releasing cytokines, which hopefully you remember are the main communication molecule. And this release of cytokines, along with the antigen presenting cell, activating all of these naive T cells. So that just basically means that they're nothing yet, they haven't been differentiated yet, they're just sitting there waiting. So once this occurs, these cytokines are going to cause a differentiation of two different cell types, our cytotoxic T cells and our T memory cells. So we're going to talk about what the cytotoxic T cells do in a second, and we already know what the T memory cells are for. So once the cells have been differentiated, we're going to have this massive expansion event happening. So that's why it's called clonal expansion. So once they have differentiated into the two different cell types, they're going to expand. So they're going to multiply over and over and over again. So we have large numbers of, especially the cytotoxic T cell, because obviously we need lots of them to perform the roles that they're going to perform. So when they have differentiated and then expanded, the cytotoxic T cells are going to leave the lymph node and they're going to eventually reach the site of infection, and they might reach an inflammation site where they're going to start playing their role. So clonal expansion just means the differentiation of those two different cell types and then the expansion, so there's lots of numbers to fight an infection. And then lastly, we're going to talk about the action of those cytotoxic T cells. So once they've been differentiated and expanded, they're going to travel to the site of infection. And we know that the T cell response is really specific to infected cells. So what the cytotoxic T cells, what their main roles are, is when they arrive at the site, they're going to, their T cell receptors are going to obviously find the infected cells and respond to the foreign antigen that the infected cells are going to be presenting on their surface. And once they've found all these abnormal cells, they're going to bind to it, and they're going to do a few things. So they can release chemicals. Some of these chemicals are called perforins, and they are going to induce apoptosis in the cell. So once the cytotoxic T cell sees which cells are infected, these perforin molecules that are released here are going to induce apoptosis. And this does a few things. So one, it kills the dead cell, it kills the infected cell, which is a good thing, so it can't replicate. And it's also going to release the virus into the extracellular environment, so it makes it easier to kill. So that's what the cell-mediated pathway does. It obviously starts off with antigen presentation and then differentiation and clonal expansion, and then our cytotoxic T cells are going to go off and they're going to induce apoptosis in our infected cells. And then our memory cells, our T memory cells, are obviously going to stay in the bloodstream, in the lymph nodes, and they're going to wait for a secondary exposure, so we can do all of this a lot quicker the second time around. We then have the humoral immune response, and this involves B lymphocytes. So cell-mediated immunity was T lymphocytes. This one is B lymphocytes. So I need to preface here that antigen presentation still happens in this pathway. So antigen presentation happens, that first step happens no matter which pathway. The T helper cell is involved in both pathways to activate both pathways, and it will be determined by what kind of pathogen needs to be defended against. So if we've got an extracellular pathogen, that's when a B cell or a humoral immune response is going to be activated by that T helper cell. That's because the main effector cell of a B cell response is the B memory cell, which produces antibodies. And antibodies are really good at targeting and marking extracellular pathogens to neutralize and basically destroy them. So if there's an extracellular pathogen like a bacteria or a parasite or something like that, we're going to initiate a humoral immune response. And then you'll see there's also 3 different cell types for this humoral immune response as well. We've got our B plasma cells, like I said, so they're responsible for generating antibodies, so that's their main purpose. We've again got our B memory cells, so they have the exact same role as T memory cells. They're going to sit and they're going to wait and they're going to provide long-term immunity if there is a second exposure. And then we have our antibodies. So our antibodies is our main effector component of the humoral response. So they're a protein that's going to be specific to the antigen. And it's going to combat pathogens in a variety of ways that we're going to go through in a second. So after antigen presentation has occurred and T helper cells have decided that a humoral response is, what we want to be initiated, we're then going to have clonal selection theory occur. So this is quite similar to clonal expansion in T cells. It just has a different name. So a B cell with a complementary receptor to an antigen will bind to it, and the B cell is said to be selected. So remember I said that B cells can also be antigen presenting cells. So a B cell can also present antigen to a T helper cell, which can speed up this process. The T helper cells that are selected through antigen presentation are also going to have complementary receptors to the same antigen because they're all very specific. And again, it's going to release those cytokines. And those cytokines are going to be responsible for clonal expansion and clonal selection. So the cytokines are going to, again, differentiate into two different cell types. So it's going to cause differentiation of naive B cells into either plasma cells or B memory cells, and then they're going to expand and make many copies of themselves. So it's really similar to the T cell clonal expansion theory. It's called clonal selection theory because B cells can act as antigen presenting cells as well, and they can also be selected by a pathogen or by a T helper cell. So once they have differentiated and expanded, this is just again going through that differentiation of those two different cell types that the T helper cells can help stimulate. So we've got our B memory cells that provide long-lasting immunity, and then we've got our plasma cells, and we're going to focus on what those plasma cells do. They create antibodies. So what antibodies are is they are a protein, which hopefully we know lots about. They have a really, really specific shape. And because of this specific shape, they can respond to specific antigens. So you'll see here, this is the structure of the antibody over here. We've got a variable region. So this is the region that's going to be different amongst antibodies depending on what the antigen is. And it's variable because it's got this antigen binding site here. And this is going to be really specific to whatever the antigen is, and it's going to change depending. So then we also have this constant region, which is going to be consistent amongst all antibodies as well. It's also split up into several different chains. So we've got our light chains in yellow, our heavy chains in blue, and then disulfide bonds that are holding all of this together. So hopefully all of this stuff sounds sort of familiar because this is just a protein that's been folded into its tertiary quaternary structures. So the B cells are going to secrete these antibodies, and they have a range of different functions, which we're going to go through in a second. They've got the specific binding sites, and there's 5 different types. So you don't need to know the different types, but a couple of key ones to probably note is IgE. So IgE is the one that's really specific to parasites, but also specific to allergic responses as well. So if there's an allergic response, and remember the antibodies bind to the mast cells in that, they're going to be IgE antibodies. And then we've got IgG, which is our most common antibody. So that's your traditional, antibody you're going to see. We've got IgA, which is specific because it can be found in breast milk and can be passed on to children. IgD, which activates other immune cells. And then we have IgM, which is normally the first type of antibodies that's produced and might differentiate into the other types. So why do we put all this effort into creating these antibodies, and what can they do? So they can perform, they're not limited to, but these are the main 5 functions that they can perform. You don't need to know, well, it's nice to know all of them in detail, but if you have a couple up your sleeve, you should be able to answer most questions knowing a couple of these in detail. So they can perform these 5 different functions. So remember, they're really good for extracellular pathogens, and these functions will kind of explain why. So they can perform something called neutralization. So this is basically where antibodies will bind to a pathogen, and they're going to block the sites that they can use to enter a cell or release a toxin or something like that. So you can see these little blue things, they're antibodies, and they've been bound onto toxins and onto the pathogen to prevent them from doing anything nasty. So that's neutralization. They're going to neutralize them. They can do something called agglutination. So what this is the antibodies and the antigens, when they bind, they can form these large antibody-antigen complexes. So you'll see here, there's obviously the antigens on a foreign red blood cell, and the antibodies are binding, and they've been bound in this big complex. So this complex makes it easier for phagocytosis and recognition. Because they're bigger, they're much more easy to recognize by phagocytes and complement and things like that. So it's much easier to destroy them. So that's what agglutination is. We've also got immobilization, which uses the same picture because immobilization just basically means to restrict the movement of pathogens. So the antibodies will bind to the antigen or the pathogen, and this will slow it down and it will stop it from moving around and spreading. We also have opsonization. So what this is when it just directly binds to the surface of a pathogen to make it easier to phagocytose specifically. And then we have activation of complement proteins. So hopefully you remember this is a component of the innate immune response, and they had a variety of functions of their own. But antibodies can also facilitate and help the formation of the membrane attack complexes, which hopefully you remember cause lysis of an infected cell. So antibodies can do a variety of things, most of them helping for extracellular pathogens. So knowing a few of those is definitely going to be beneficial. And let's just dive a little bit deeper into immunological memory because it's going to tie into what we learn about in chapter 8. So that you'll notice that both B and T cells differentiate into memory cells, and these are going to remain in the blood for extended periods of time. And they're going to allow the body to recognize the pathogens it's previously encountered. So that's what makes it adaptive. So we're not born with it. It develops over time depending on the types of pathogens that you are exposed to. And this is going to ensure that we can quickly and effectively respond to this pathogen again the second time. So if you get a cold or something, and then you get infected with that cold again, you're probably going to be sick for a lot less time the second time. And this is also the concept that vaccines work around, which we'll go through in chapter 8. B memory cells, once they're activated that second time, what they do is they rapidly divide into plasma cells, which produce large amounts of antibody. And because they can do this so quickly and they don't have to go through clonal selection and all of that sort of stuff, they can really quickly and easily create a bunch of antibodies, which can immediately knock out the pathogen. T memory cells are very similar, so they'll rapidly divide into our cytotoxic T cells upon antigen presentation, and again, they can very quickly, without clonal selection, expansion, whatever, they can very quickly go about their job and induce apoptosis in infected cells. So this is really good and really helps us to prevent severe illnesses. So this is just a summary of everything we've gone through. I know it is a lot, but in summary, if our first and our second lines fail, we bring in this really specific, really unique, really tailored response to hopefully get rid of this pathogen. And assuming that this pathogen is quite a bad one because it's evaded our first two responses, we can also create memory towards that pathogen, which is going to mean that we get less sick the second time we're exposed to it and times thereafter. So this summary diagram just splits it up into two different types. So we've got our humoral response and our, we've got our humoral response and we've got our cell-mediated response. So remember, our humoral response is better for our extracellular pathogens, and our cell-mediated response is better for our intracellular pathogens, just based off what their effector functions are. So despite which pathway we have to go through, either way we're going to have this antigen presentation. This is normally going to be a dendritic cell, and it's going to activate a T helper cell that's specific, and that T helper cell is then going to release cytokines. And depending on what the pathogen is, it's either going to release cytokines to create a humoral response and activate B cells, or it's going to produce cytokines to activate a T cell response and produce cell-mediated immunity. So in the B cell response, we've got our two different differentiated cell types that can occur after clonal selection and differentiation. We've got our long-living memory cells, which I said really important for second exposure, and we've got our plasma cells. And these plasma cells' main job is to secrete antibodies, which have a range of effector functions that help to eliminate extracellular pathogens. If it's an intracellular pathogen, again, the T helper cells are going to release cytokines, and our naive T cells are going to start differentiating into two types, our memory cells that we know and our cytotoxic T cells. And they're our main effector cell for this one because they're going to go around and they're going to release perforins, which are going to induce apoptosis in infected cells, which will hopefully reduce infection. So I hope this helps. If you have any questions, bring them to class and I will see you there.